Hello, everyone. Welcome to the Environmental Research webinar for this 2025 and 26 new semester. So my name is Sungkyun Park, and I'm a faculty member in the Department of Epidemiology and EHS. I'm very delighted to see returning colleagues and to welcome new students and center members joining this webinar first time. So this seminar series is sponsored by the Michigan Life Stage Environmental Exposure and Disease Center, also known as M-LEEaD. So I'm very excited to introduce this year's first speaker, Dr. Lidia Mínguez Alarcón. Dr. Mínguez Alarcón is a Spanish epidemiologist, and her research focuses on identifying chemical and non-chemical stressors of human fertility, reproductive and cardiometabolic health, with a special focus on diet, chemical interaction, paternal contributions to pregnancy outcomes, and chemical mixture. Dr. Mínguez is currently an assistant professor of medicine and a lead investigator at Harvard Medical School at Brigham and Women's Hospital. Dr. Mínguez received her Bachelor of Pharmacology from the University of Valencia School of Pharmacy and her MPH and PhD from the University of Murcia School of Medicine. I'm sorry if I mispronounced the name. She previously completed a postdoctoral fellowship in the Department of Environmental Health at the Harvard T.H. School of Public Health. Please join me in welcoming Dr. Lidia Mínguez Alarcón. Lidia, your podium is yours.
All right. Thank you so much for the invitation to present today, Sam. I'm so happy to be here with all of you to share part of the research that we have conducted during the past years at Harvard using data from the Earth study on chemical, non-chemical stressors and human reproductive health. So first, I would like to start this talk acknowledging I'm very proud to be part of this fantastic team. Not one single paper will be possible to publish without any of these team members. All the study participants, the NIH funding, and for those who do not know, this team is led by Professor Russ Hauser at HSPH, and he's the PI of the Earth study. And I would like also to take this opportunity to thank Russ for his amazing leadership in the Earth study. So let's talk a little bit about Earth. The Environment and Reproductive Health Study is a prospective cohort study that explores environmental, dietary, and other lifestyle factors in relation to fertility, pregnancy, and offspring endpoints in couples attending the MGH Fertility Center in Boston. So this study cohort was established in 2004 and ended in 2021. Over 1,000 women and 500 male partners, they were enrolled in the Earth study. So to quantify chemical concentrations, we collected thousands of urine samples from study participants during pregnancy for female partners and before conception for both male partners and women. Female partners. So we evaluated ovarian reserve in women as part of the initial infertility evaluation, and in men they provided semen samples in each fertility treatment. So couples, they underwent different types of treatments, either IVF or IUI, and a few couples, I'll say around 15%, they got pregnant, the female partner got pregnant naturally during the course of the study without any medical treatment. So a little bit of background. As many of you know, probably, infertility is defined as the inability to achieve a pregnancy after several months of attempted conception, and it is estimated to affect between 13 and 18% of all couples worldwide. And male factor, which is defined according to the WHO reference values for semen quality, has declined over the years, and it is considered the underlying or contributing cause of infertility in around 40% of the cases. So given this, there has been a considerable increase in the use of medical treatment among couples with decreased fecundity. And fertility rates, as you can see, due to assisted reproductive technologies have also increased, especially in the US. However, not all couples trying to conceive, they can afford or have access to these techniques to conceive. So there has been like a great interest in identifying modifiable factors like chemical exposures that can predict human fertility during the past few decades. So chemicals of interest include EDCs, endocrine disrupting chemicals. These are chemicals that they can act as fake hormones and they can interfere with the endocrine system. And they can be found in numerous consumer and personal care products. And humans, they are exposed mainly through inhalation, ingestion, and transdermal absorption. Some examples, as many of you know, phthalates, bisphenols, parabens, triclosan, pesticides, flame retardants. And one of the biggest problems related with them, or the major problem, is that there is little regulation in their use, as nearly all the US residents, they have detectable concentrations in their urine. Exposure to these chemicals, they have been associated with several health effects, both in humans and animals, and for example, in the reproductive system, causing infertility. Earth, as I already mentioned, was designed to evaluate exposure to these environmental factors in relation to reproductive health and fertility problems. And lately, we have focused on addressing novel questions, specifically how diet might modify the associations between EDCs and reproductive outcomes. Second, clarifying the paternal role of exposure to these chemicals on couples' reproductive outcomes, including pregnancy, probabilities of pregnancy and live birth. And also assessing these chemicals as a mixtures, applying new approaches that they were recently developed. Since humans, we are exposed to hundreds of chemicals at the same time and not only one. So today I'll summarize some papers we have published in Earth during the past few years with a special focus on these three areas. So exposure to some of these EDCs, they have been shown to promote obesity, and they are called obesogens. Three mechanisms of action, they are postulated as the most important ones by which obesogens, they exert their effects. One is altering the adipocyte development, and second is interfering with hormone receptors, and the third one is promoting inflammation, as you can see in the slide. So I will start with this part of the talk on diet-chemical interactions. So an interaction between a dietary factor or nutrient and chemical is possible because they both might be present in the same food. They can share internal mechanisms of transport, metabolism, and cellular uptake, or target the same molecular signaling pathways and biological functions. So traditionally, study cohorts, they were designed to explore either exposure to environmental chemicals, or intake of nutrients or food. So Earth started in 2004, but in 2007, we also started collecting a diet info using a food frequency questionnaire that allowed us to conduct the analysis that I'll show now. So this is a paper on BPA, folate intake, and repro outcomes in Earth. So for this paper, Experimental data in rodents suggested that the effects of BPA on oocyte development, they might be modified by dietary methyl donors. So we wanted to evaluate whether the same interaction was true in humans, because it was unknown. So we evaluated whether intake of methyl donors, specifically folate, vitamin B12, choline, and betaine, they modified the associations between BPA concentrations measured in urine samples and pregnancy outcomes among women in the EARTH study. So to do this, we first divided women in two groups by median intakes of these methyl donors, and specifically 400 micrograms per day of food folate. That, by coincidence, is actually what is recommended for intakes for pregnant women. And we found that high urinary BPA concentrations, they were associated with lower probability of implantation, clinical pregnancy, and also live birth among women who consume less than 400 micrograms per day of food folate, but not among women who consume higher intakes. So, suggesting a protective effect of folate intake against the detrimental effect of BPA on repro outcomes, but only at the higher levels. Our results, they were only significant for women in quartile 4 of BPA exposure, as you can see in the figure. So BPA is an endocrine disruptor that binds with diverse estrogen receptors and can mediate the activities of endogenous estrogens. And there is also evidence that the folic acid metabolism impacts ovarian responsiveness to gonadotropins, as well as the basal and stimulated production of estradiol in the granulosa cell, suggesting potentially an unknown mechanism linking folic acid metabolism with the production of estrogens in the ovary. So an interaction between folate and BPA is possible, and again, and I think this is something super important from the talk that people, they should take away. Exposure to this and others EDC is universal, and humans, they have little control of it. So results from this study can translate into low-cost clinical interventions, since advising women on intake of folate may be potentially used to protect against the negative repro effects of BPA. So this is a similar paper. We explored something similar, but in this case, the stratification was made by soy intake. So in this case, what motivated us to perform this analysis was a paper in mice showing that BPA was related to oocyte abnormalities in animals, that they were fed with a phytoestrogen-free diet, while no effects they were seen in mice fed with a soy-based diet. So again, we stratified women, but in this case was soy intake, yes versus no. And we found similar results as for the folate BPA paper, but in this case, we observe a trend. Specifically, soy food intake modified the association of urinary BPA concentrations with live birth rates. Among women who did not consume soy foods, the adjusted live birth rates per cycle in increasing quartiles of urinary BPA concentrations, they were 54%, 35%, 31%, and 17%. And no associations that were observed for women who did actually consume soy foods, as you can see in the right part of the figure. So these results, they suggest that soy food intake might protect against the adverse reproductive effects of BPA. And this was the first report suggesting a potential interaction between soy and BPA in humans. However, I feel like every time that we publish a paper, we come up with a different problem or with a new question. So the main problem of these two papers is like data on food intakes that were assessed using a food frequency questionnaire. So misclassification of the exposure is it's possible. So we published this paper recently, and we try to address this limitation on this paper on urinary phthalates, omega-3 measured in serum samples and pregnancy outcomes in women. So there is substantial evidence from systematic reviews and meta-analysis and of trials indicating that supplementations of omega-3 during pregnancy, they are associated, or they can have numerous benefits for pregnancy, not only for the pregnant mother, but also for the offspring, including the reduction in the frequency of preterm birth, possibly extending a gestational length, reducing the frequency of preeclampsia, improving metabolic parameters in pregnancies affected by diabetes, etc. And on the contrary, exposure to phthalates have been negatively associated with most of these outcomes, as you probably already know. And also, a biological interaction between PFAS and phthalates on pregnancy outcomes is possible because both PFAS and phthalates, they can be bind to PPAR receptors, including PPAR gamma, which is known to be involved in placentation as well as embryonic and fetal development. So for this analysis, we try to evaluate the effect modification of serum PFAS levels on the associations between urinary concentrations of phthalates, biomarkers, individually and also as a mixture on pregnancy outcomes among women in Earth. And we found that concentrations of the phthalate biomarkers mixtures, they were associated with higher adjusted probabilities of pregnancy loss and lower probabilities of live birth among women in the lowest tertiles of some type of these PFAS, but not among women in the middle or high serum concentrations of these PFAS. So this study suggests adverse effects of the phthalates on pregnancy loss and live birth may be attenuated by intakes of some type of omega-3 fatty acids. And these results, they could inform clinical practice, reducing the burden of infertility by these chemicals, by phthalate exposures among the general population, and improving pregnancy outcomes among subfertile couples. So this is the second topic that I'll talk today, is on chemical mixtures. So most of the epi literature on chemical effects on human health, they have focused on evaluating chemicals individually, which is actually very helpful to find potential mechanisms explaining the associations. However, humans, they are exposed to thousands of chemicals at the same time. So evaluating chemicals as a mixture has emerged to account for potential correlation and between the chemicals, and also better represent real-life chemical exposures. So this is a paper that we published a few years ago, and we investigated the association between concentrations of parabens, again measured in urine samples, and glucose levels during pregnancy and among women who participated in the Earth study. So we evaluated the chemicals individually and also as a mixture, and we found that urinary concentrations of butylparaben, the concentrations, they were positively associated with blood glucose levels. Also, when assessed individually and also when assessed as a mixture, applying BKMR, which accounts for correlations between the two other parabens that we also consider for this analysis. And we also found an inverse associations between urinary propylparaben concentrations and glucose levels. Again, when assessing the specific paraben individually and also as a mixture when accounting for correlation and residual confounding by the other two parabens. So some mechanisms that explain the associations, there could be several. First, parabens, they can activate glucocorticoid receptors. So they can somehow interfere with normal glucose regulation. And second, parabens, they could also act as thyroid hormone receptors and could alter normal thyroid functioning. And third, parabens, they are also agonists of PPAR receptors, which are involved, as I already mentioned, in glucose metabolism. And in this other paper, we applied two methods, principal component analysis, which groups the chemicals based on the correlations and creates different components. And also we applied BKMR, which accounts for correlation, evaluates individual chemicals accounting for correlations for the others, and also can tell you what is the association between the whole mixture of the chemicals in relation to the outcome. So this was a cross-sectional analysis, and we evaluate the mixtures of urinary phthalate metabolites in relation to thyroid hormone levels during the preconception window among women in Earth. And what we found is, using principal component analysis, we found that mixtures of urinary DHP metabolites, it was one component that it was created when running the PCA, was inversely associated with the serum biomarkers, some serum biomarkers of thyroid function, some thyroid hormones. And also applying BKMR, we observed that one specific phthalate was the primary contributor to the negative associations, was basically driving these negative associations. So we found that these results, they were particularly important since they provide evidence that humans, they are exposed to a group of chemicals at the same time, first, and also associations of phthalate mixtures with thyroid hormones among women that they are trying to conceive during the preconception period. And then now I'll move to the third topic, the paternal contribution. So as I wrote in the slide, the EP literature has linked male environmental exposures with sperm DNA mutations, and some evidence from experimental animal studies has shown that paternal environmental exposures, they might affect offspring health via epigenetic alterations in the sperm. So the problem with traditional study cohorts evaluating chemical exposures in relation to pregnancy outcomes or offspring health is that enrollment to evaluate these pregnancy outcomes or even perinatal was during pregnancy. So there were only a few cohorts that enrollment was during preconception, before pregnancy. So taking advantage of the EARTH study, that is a preconception study cohort in which we enroll not only moms, but also the male partners during preconception, we were able to evaluate some paternal exposure to chemicals in relation to couples' pregnancy outcomes. But first, I will start with this paper on semen quality and exposure to BPS. So we have also investigated repro effects of emerging and understudied endocrine disrupting chemicals. Because of new regulations, exposure to some chemicals is decreasing, whereas exposure to new replacements like BPS is increasing. So these chemicals, they were introduced in the market without proving their safety first. And I have to say that we detected BPS, which is again a BPA replacement in 76% of the men. And we found associations of urinary BPA concentrations with lower volume, sperm concentration, total count, and motility in models that they were adjusted for urinary BPA concentrations as a potential residual confounding. And we found stronger associations among obese men. So one possible explanation for these interesting findings is that overweight or obese men, they may be more sensitive to BPA exposure, given that they are at the same time exposed to our hyperestrogenic environment, since obesity increases circulating estrogen levels in men, and also anti-androgenic signal, since BPS is anti-androgenic, has an anti-androgenic activity. So this was the first paper investigating repro effects of this new chemical, not that new anymore, BPS, and showing its detrimental male reproductive effects among men in the EARTH study. However, whether the potential effect of certain chemicals on decreased semen quality has any consequences on the female partner pregnancy outcomes is not that well known, as most of the literature on pregnancy outcomes, as I already said a couple of minutes ago, has focused on female exposures. So to address some of these questions, we conducted this study to evaluate whether mixtures of paternal urinary BPA, parabens, and phthalates altogether, they were associated with pregnancy outcomes in the female partner among couples in EARTH. Again, paternal or male exposures in relation to female partner pregnancy outcomes. And it was very interesting what we found. We observed that paternal exposure to some phthalates, not the other chemicals, some phthalates, biomarkers, they were associated with higher probability of couples infertility treatment failure with a hazard ratio of 1.4. And the most interesting part is like results remain stable or significant. The associations remain even when accounting for the female partner exposures that they were actually highly correlated between them, the female and the male partner chemical exposures. So these results, what they indicate is that the paternal exposure to some phthalates is negatively associated with the probability of having a live birth in the partner. And they also highlight the importance of considering paternal chemical exposures when we are investigating environmental exposures on couples' reproductive health outcomes. What we need is other studies confirming or not these results, and super important, other studies evaluating the potential biological mechanisms or pathways that explain the associations between male exposures and female partner reproductive outcomes. As I mentioned at the beginning of the talk, every time that we publish a paper, we come up with another question. And I think this is in part what we are doing next. So in this paper that is under review, and what we are trying to examine is the associations between urinary phthalate metabolite concentrations, metabolic perturbations or metabolomics measured in serum samples using untarget metabolomics in men, and we are also evaluating whether these metabolic pathways, they are associated with the probability of live birth among this couple. So far, what we found is that urinary phthalate metabolites in men and live birth in the female partner, they were mostly associated with serum pathways involved in lipid, amino acids, DNA, and carbohydrate metabolisms. So these results, they provide new insights into potential, as I say, potential biological mechanisms linking male phthalates in relation to live birth among couples seeking fertility care. So what is next? We're busy. We are currently working in these two other ones from NIHS. So the first one is on male pesticides, epigenetics, and repro health. So we will be quantifying pesticide concentrations in urines from men and women in the EARTH study and looking at associations between them and pregnancy outcomes. And we just finished, we are also conducting epigenetic analysis in both blood and sperm male samples to trying to find potential mechanisms that explain the associations between these male pesticides and female partner pregnancy outcomes. So this project is in collaboration with Andres Cardenas at Stanford and Rick Pelsner in Wayne State University. And in the second other one, we are quantifying phthalates and flame retardants in urine samples and evaluating associations with cardiovascular and metabolic health in midlife. So this is a study cohort that it's named LEED, longitudinal investigation of health and diseases after infertility. And our goal is to re-enroll women that they participated years ago in EARTH during reproductive years and to evaluate their cardiovascular and metabolic health in midlife. So most women, they are now perimenopausal, so they are at high risk of cardiovascular disease. And this study, real quick, is conducted in 3 steps and remotely. So the first day, women, they are collecting a urine sample and completing surveys. The second part of the study, in day 6, they have to collect another urine. And day 7, a week after they enroll, we have a phlebotomist coming to their homes to collect blood and to do some blood work, and also collect some anthropometry measures. So our goals are to investigate preconception, prenatal, and long-term urinary concentrations of phthalates and flame retardants in relation to cardiovascular and metabolic health. So these chemicals, they can be bind to PPAR receptors, which are related, as I already mentioned, to glucose and lipid metabolism, as well as they are involved in inflammation. We would like also to identify the most important window of exposure that predicts the outcomes, and which can be done with LKMR, which is a novel method to evaluate mixtures at different time points. And also would like to explore changes in cardiometabolic health by chemical exposures before and during pregnancy. Also, fertility measures like AMH levels, infertility type, pregnancy outcomes seem to be predictors of future cardiovascular health. So if we find associations between these chemicals and the outcomes, we will evaluate whether the associations, they are mediated in part or in part explained by these important fertility measures. And the goal of this R1 is to demonstrate that women who are highly exposed to these chemicals during repro years are not only at high risk for infertility, but also at high risk for cardiovascular and metabolic disease later in life. And I think that's all, everything that I wanted to say today. I don't know if we're on time for some questions.
Yeah, great. This is very interesting, and a lot of data you show us, and maybe the audience need time to digest the finding, and then asking question, and so please use Q&A menu on the bottom. You can see that, and then enter your question, and we will take care of the question. And So Lydia, I don't know if you can see the question, but I will read the question, and you can also you can open the question from the Q&A, and then you understand what is asked. So the first question we receive is that inspired by your plan to study exposure on cardiovascular health in midlife, are there findings of phthalates and other chemicals affecting reproductive health in aspects other than conception and birth? For example, more painful menstrual cramps, irregular cycle, early late menopause, and etc.?
Yeah, so we are actually collecting information on we are using the menopause scale to collect information on menopausal symptoms and also age and natural menopause. So yes, we would like to do that, but probably in the future. The first thing is we need to do is accomplish the aims on glucose, insulin, lipids, inflammatory biomarkers. But yes, we are collecting information on menopausal symptoms at midlife and age and natural menopause to potentially work on 1 or 2 papers in the future evaluating that.
So right now in Earth, both Earth and LEED, I forgot the acronym of the second cohort. LEED. And then, how old they are currently? So you are recruiting them again, and then they are in the now midlife, and they are on the
they are
menopause, right?
Yeah, we were trying to reach out to women that they were already 45, but we went down a little bit to 43. So I think between 43 and 50 something.
Okay.
The ones that we recruit. And
then, so how do you you're planning to determine the menopause within that study? So are you using some a menstrual diary to identify and determine the timing of the menopause within those studies?
Yeah, so it's a question. It's a question in the questionnaire. It's like, did you reach out menopause? Yes or not? Yes. How old were you when you reach out menopause? So.
Okay. All right. So we have another question from our recent graduate and currently postdoc, Dr. Sonyoung Park. So what's the chemical exposure measured in midlife as well? So you measure chemical exposure in midlife. And I think I'm curious if there are some patterns when comparing gestational exposure versus midlife exposure. And if so, how would this be adjusted in the current studies?
Yeah, so we are actually, one of our goals is to try to calculate trends from preconception, pregnancy, and midlife in these chemicals. Some of these women, they enroll in Earth 20 years ago. So my guess is like, for example, 20 years ago, they had very high levels of BPA and low levels of BPS and BPF. And it'll be the opposite nowadays when they enroll in LEED. We are expecting to see low levels of BPA and high levels of BPS and BPF. So with the mixers methods that we want to use, the LKMR is a very novel method that you evaluate windows of exposure when adjusting for the other 2 windows. So I think that will somehow address the limitation of residual confounding by another window.
Maybe the related question that the right before you introduce your new study looking at this midlife, and that you show that the paternal contribution, and which I found very interesting, and then you mentioned that this paternal exposure, even after you adjust for the maternal exposure, you see the association. But did you also see association with the maternal exposure, and the maternal exposure effect is even stronger? Or so which one is
this is the funny thing. The funny thing is, like, we did first the maternal paper, And we found borderline associations, very weak. Then we did the paternal paper, and we found negative associations with I think it was the DHP metabolites. And the results, they remain after adjusting for female exposure, that we already knew they were going to, it was not going to change, because we first published a paper with maternal exposures, and we found very weak associations. So it is actually super interesting.
Very interesting. Okay, so
we're expecting to see something with the male sperm epigenetics, or male blood epigenetics. Otherwise, we don't know how we are going to explain those results.
Yeah, I think it's challenging to interpret why. Some discrepancy between the maternal exposure and the paternal exposure. And then, as you mentioned, that like epigenetic pathway might be one possible explanation, although maybe this is an important next step you understand.
Especially because the chemicals, at least in our study cohort, these chemicals, they are highly correlated between couples, the maternals and the paternal exposures. So it's important to take each other into consideration as well.
Right.
Not only because you are evaluating the female outcomes, the pregnancy outcomes, but also because they are highly correlated.
Yeah. Okay. Yeah. Super cool. Maybe I can add a couple more questions, and then because early your presentation you talk about like a dietary, like a diet and chemical interaction, which I found very interesting, and this is really important, especially for chemical, because the chemical is it's not easy to like a control. And then
we are publishing nowadays on mercury and omega-3. We've got these results on serum omega-3 from a pilot study, and we are publishing a little bit also on mercury and omega-3, because they both, in the general population, the main source is fish intake.
Right. Yeah. So but within your study, you found that.
And then
BPA and what was it? The BPA folate and the BPA in the soy intake, and then so then are you expecting, or your interpretation about that interaction is that like the soy is also phytoestrogen, so that's why the soy intake is a more estrogenic effect.
Yeah.
And then, your hypothesis is that the BPA is a more anti-estrogenic activity, and then so that's why the soy phytoestrogen counteract that anti-estrogenic effect, and so that is your. Okay. So
and they can also compete with the receptors. So it depends on the nutrient that you are evaluating. In this case, they can compete with the receptors. In the case of the fish, for example, the mercury and the omega-3 is because both they are present in the same food. So there is a residual confounding by the other compound. So it depends the type of nutrient that you are evaluating.
But what about folate? Folate is a totally different, the dietary component, and then maybe the biological mechanism could be different, and then the one carbon metabolism, and maybe that is how regulating this the estrogenic effect is. So that is how the folate is working.
Correct. Yeah. Okay. And it's actually pretty cool, because to see the animal papers and to try to not replicate, but try to do something like similar in humans. And folate, I mean, there are plenty of studies showing the benefits not only for the moms during pregnancy, but also the offspring.
So then you are using the 400 is a milligram per day or microgram?
Microgram. Sorry, I didn't say milligram. Micro. So, but that by chance is actually what is recommended, and by coincidence, it was close to the median in our study population.
Okay, so then as a kind of recommendation to the the general audience.
Yeah, and I would like to say something that actually I forgot. We found an interaction with food folate, but we didn't find any interaction with supplemental folate.
Ah, okay.
And we didn't find any interaction with supplemental folate. We think it is because these women, they are trying to conceive, so they take supplements, and they have very, very high levels of folate from supplements.
Okay.
So all of them, they are in one side of the distribution. So it's very difficult to divide women into two groups to try to evaluate interactions or stratification by median supplemental folate.
Okay. Yeah. So I have a couple of more questions, but we have a couple of other questions. So let me go from the bottom. So thank you for the presentation. I have a curiosity. Does the chemical fingerprint of babies who are breastfeeding for more than 18 months differ from that of newborns who are only nursing for 6 months?
I don't know. I don't know. We didn't do follow up after birth, so I cannot answer that question.
Yeah.
We then evaluate. So the endpoint of the study in Earth was like birth, and then couples, they were done in the study. So we didn't do any follow up after birth. I think that is a very cool question, but I cannot answer.
Okay. And then the last question I see is that, so have you considered omega-3, omega-6 ratio? I think a couple of studies suggest the role of imbalance in inflammation metabolism.
We didn't, but I agree with that, and I think one of my postdocs suggested to run that. So I think we didn't do it yet, but we are planning. But we will do it. Yes, I agree.
Okay, and then so your study, so you measure this PUFA in serum. Do you see also the similar this counteract of this dietary omega-3 fatty acid intake when you are using food frequency questionnaire, not the serum concentration?
Yeah, we find similar patterns, and actually the FFQ does a pretty good job. It's highly correlated with the serum, with the levels in serum. So ideally, I mean, you want to have biomarkers of exposure, omega-3 in serum. But if funding is limited or there are some funding restriction, nowadays everything is super crazy. So using the FFQ is a very good first step.
Good. Okay. Yeah, I think we, like in this audience, there should be several other like a cohort study that can think about this potential diet and chemical interaction to find or replicate. The first step is replicate your finding, but also can find a good like a dietary intervention to mitigate this potential chemical toxicity through the intake of this omega-3 fish consumption and folate and soy product, which are very interesting. Okay, so now 12:50, and I don't see other questions. So let's wrap up, and thank you for your interesting presentation and also this Q&A.
Thank you so much for the invitation.
So for the audience, so we will have a our webinar. So Meredith put the next webinar series, and but this semester we have a couple of interesting other seminar lined up, and also I will email later that we plan to do more. The work in progress seminar with the current doctor or postdoc trainee, and then having more informal, the conversation about the current research within University of Michigan. So please stay tuned, and thank you for attending this webinar. And again, thank you, Lidia.
Thank you so much for having me today. It was my pleasure.